Hi guys, I'm Ethan Rydell, and today we're going to be looking at the phases of meiosis. The first and longest phase is interphase. And what we start with is a beginning cell, which has two pairs of chromosomes, first pair and second pair. What happens in interphase is, first of all, the centromeres duplicate. And you see, you have these DNA that are not tightly loosed into a chromosome, it's all kind of jumbled. So what interphase does is, first of all, it duplicates the DNA and winds it up into tight chromosomes. You see, this is one set of DNA and two sets of DNA. Duplicated it. Now, in prophase 1, we undergo this thing called synopsis, when the male chromosome and the female chromosome cross over, as we see right here. We do this to create variation in the offspring. You can see parts of the female DNA is mixed with the male DNA, and parts of the male DNA is mixed with the female DNA. We do this because instead of just having the male chromosome and the female chromosome going to the kid, this creates a whole new chromosome for the offspring. Now, in metaphase, the chromosomes line up in the middle along what is called the metaphase plate. Now, the chromosomes could align themselves with the blue on the left side and the red on the right side, but they could also did it the other way. Same thing for here, and you can have 4 different combinations with this. That's represented here. That is just giving us more variation. Now, when you think about it, we have 23 chromosomes for our gamete cell. So, in total, we're going to have a lot of different combinations, just giving us more and more variation for our offspring. And that is represented as independent orientation, right here. Now, for the rest of metaphase 1, we see the spindles, which are in the middle of the chromosomes, attaching them, connect to the centromeres lines right here. Now we see in anaphase 1, the 2 chromosomes get pulled to either side, preparing to go into 2 different cells. Now what we do in telophase 1 is we're reforming 2 nuclei, getting ready to divide the whole cell altogether. Directly following telophase 1 is cytokinesis, where the 2 nuclei get separated into 2 new cells, ending meiosis 1. Now we see that once we have 2 new cells, prophase 2 can start. We're not going to cross over now like we did in prophase 1, just because we've already crossed over. We have variation. So now it's going to turn, line up along the middle. Now metaphase 2 goes hand in hand. See, the spindle fibers again connect with the centromere. See, I highlighted it to show you guys. Right there. Now in anaphase 2, each of the chromosomes are being pulled to another side. And just to clarify with you guys, the reason why we're having a second anaphase and a second metaphase is because, unlike mitosis, we're making 4 new cells here, not 2. So we need to do this a second time to duplicate it again. And now finally, during telophase 2 and cytokinesis, we've created 4 different gamete cells. Now, if we look back to where we started, we had 1 cell with 4 chromosomes. Now we have 4 different cells with 2 chromosomes. The reason why we have half the amount of chromosomes for each cell is we're making sperm cells if you're a male. So this can fertilize with an ova or an egg and 23 chromosomes. Now, obviously, we can't do 23, so we're doing 2. But if this was a real human, it'd be 23 chromosomes fertilizing with an egg. That would become 46 chromosomes, a full body cell. Thanks, guys. It's been fun. And just a little side note: if your kid asks you where babies come from, don't go this much into detail. See you guys later.